Welcome back, class 9 to the online bio classes. In the previous class, we had already studied chapter 4, but we had not completed the exercises. I will try to give the answers if we get the time at the end. But today we will start with the new chapter 5, structure and germination of seeds. You know what is a seed? Seed is a ripened ovule or a mature ovule which is formed after fertilization, and it is present inside the fruit, and fruits are formed from the ovary. It is the ripened ovary which changes into a fruit, and the ripened ovule develops into a seed. And depending on the number of cotyledons, you know that cotyledons are the one which store food inside a seed. It contains different types of food materials like fats, proteins, carbohydrates, and inside the seed we have a baby plant which is present, and the baby plant is present in the form of the embryo that after germination gives rise to the root system, is formed from the radicle, shoot system develops from the plumule, and the food is stored for the germination inside the cotyledons. Then you know that the seed is protected by a hard outermost covering which is called seed coat, which has two layers. The outer layer is called testa, the inner layer is called tegmen. Now next we have to study is types of seeds. There are two types of seeds. What are they? Depending on the presence or absence of cotyledons. First one is, if there are, depending on the number of cotyledons, there are two types of seeds. Like if only one cotyledon is there, only one cotyledon, such seeds are called monocots. And if the number of cotyledons are two, two cotyledons are present in a seed, they are called dicot seeds. The name is given dicotyledonous or dicots. Now next is based on the presence or absence of endosperm. What is that endosperm? Endosperm is produced, in the previous chapter we had done that when one of the male gamete fuses with the secondary nucleus, it forms endosperm. It also stores food and it is present in large amount in the seeds. Now depending on the presence or absence of endosperm, the seeds are divided into two types. If the seeds contain endosperm, like the seeds are of two types, what are they? Based on the presence or absence of endosperm, it is called endospermic seed or albuminous seed. Endospermic seed. The other name is albuminous, albuminous, albuminous. And the other one is non-endospermic, or it is also known as X-albuminous, non-endospermic, or also known as X-albuminous seed. X-albuminous seeds. So what is the X-albuminous seeds? Seeds without endosperm. This is with endosperm. The seeds with endosperm, whereas these are the seeds without endosperm present in them. Means they have no endosperm present in it. That's why they are called non-endospermic or X-albuminous. Both monocot and dicot seeds can be albuminous or X-albuminous. But in endospermic seeds, like this endospermic seeds, the one which contain endosperm, which are also known as albuminous, the cotyledons are usually small and less developed, whereas in non-endospermic seeds, the cotyledons are large and they are highly developed. So here the cotyledons are highly developed. Cotyledons are highly developed, and here the cotyledons are small or less developed. Cotyledons are small and less developed. So you can see in your book also, page number 44, two different types of seeds. They are cotyledons. On the basis of cotyledons, two types of seeds are monocot and dicot. On the basis of endosperm, the two types of seeds are endospermic and non-endospermic, which are also known as albuminous or X-albuminous. The endospermic seeds contains small cotyledons, whereas non-endospermic seeds the cotyledons are highly developed and they are bigger in size. So now next we have to study about one of the seed, monocot seed, that is maize, and the other one that is dicot seed, which is the bean seed. So first we will study about the maize seed. Maize seed is also known as, actually it is a fruit, not a seed. And since the wall of the fruit in maize is fused with the wall of the seed, that's why seed also has a protective covering. So seed coat and the fruit wall both are fused together in case of maize. So that's why it is called maize grain. It's better to use the word grain instead of writing maize seed or maize fruit. It is better to say it as maize grain, because it is a one-seeded fruit which is called caryopsis. Actually it is a fruit in which the fruit wall and seed coat are fused together. So it is a fruit. Maize is a fruit with seed coat and fruit wall fused together. Fused together. Okay, now next, what are the different parts present in a maize seed? If you see the diagram which is there in your book, the diagram of a seed, maize seed has been shown. Okay, in which you can see the top scar of style is shown. SCAR. Scar means mark of style. Style, you know, is the part of female reproductive part, pistil, that is stigma, style and ovaries. So scar of style is shown. Then you can see that the endosperm is marked here. Endosperm is the stored form of food which is present in the seed. Then the next one we have is the position of embryo, which is shown. Embryo is the baby plant which is present. Position of embryo. So embryo means the baby plant that is present inside the seed that germinates to give rise to the root system and the shoot system. Similarly, you can see the position of cotyledons, and there is a scar which marks, it is the scar of attachment of cob. Means that is the place where it was attached with the cob. Cob means in which it was present inside the female reproductive, that from which we extract all the grains are present. So that is called the cob. Understood? The maize cob, in which the grains are fitted together. So that one shows the scar of attachment of seed with the cob. Now next diagram which is there is the entire grain is shown, and the grain in LS is shown, that is longitudinal section. So that diagram is also nice. You can see, you can practice and draw it, because it will help you to mark all the parts or the level, all the parts clearly. You can see there are two distinct regions marked here. This upper region, and then there are two layers also. The outermost layer is called alluron layer, and this one is called endosperm, the one which is present here. One covering is present around it, that is known as alluron layer. Can you see in your book it is given there already? So you can see that seed coat and fruit wall, both of them are fused together. Seed coat plus fruit wall, they are fused together. So that's why it is known as a grain. Then we have the alluron layer, that is the third layer, is alluron layer. You can see it clearly in the diagram. Alluron layer is there. After that, cotyledons are present, and here you can see this one shows what is the position. It is known as plumule. Plumule is located here, and this radicle is located here. This one you can mark it as radicle, which gives rise to the root system, and this one you can mark it as plumule, that gives rise to the shoot system. This one you can mark, okay? Not this point. This one I have done like this. This one you can mark it as plumule. And then what is coleoptile and coleorhiza? Like if I have marked like this, a protective sheath around this plumule, this protective sheath is called coleoptile. So if asked what is coleoptile, a protective covering present around the plumule. Similarly, there is a protective covering present around the radicle, that is known as coleorhiza. RHIZA. So what is coleorhiza? a protective sheath present around the radicle. What is coleoptile? A protective sheath or covering which is present around the plumule. And then what is present in this part of the seed? This lower part of the seed contains cotyledon, cotyledons. Since only one cotyledon is there, it is a monocot seed. The upper region contains endosperm, which is common in both the diagrams shown here, and this one's the lower region contains cotyledons, and in that region only you find the embryo, which contains plumule and the radicle. Plumule gives rise to the shoot system, radicle gives rise to the root system. So that part shows the embryo. Okay. Now, what are the different parts of a maize seed? Number one is a seed coat. It is a brownish membranous layer which is made up of seed coat and the wall of the fruit, which are fused together. It is a protective layer. So three layers are there, as I told you. This is the third layer I am going to draw. Okay. So seed coat. This one, suppose outermost seed coat, this is the fruit wall. Both of them are fused together. These two are fused together. Understood? So that's why it is known as a grain. Then comes endosperm. Endosperm is the food storage tissue. It is forming the main part of the grain, and it is separated from the embryo by a layer of thin epithelium. So you can see this one is the epithelial tissue which is present. So if you want to mark it, you can mark it as epithelial layer. Epithelial tissue you have studied. Epithelial is the name of the type of animal tissue, but here it is there in the plants also. Epithelial layer is the one which separates the upper region, that is the endosperm, from the cotyledons. And then the embryo consists of a single cotyledon which is known as scutellum. So what is scutellum? It is the name given to the cotyledon part of the maize seed. It is called scutellum. scutellum. Only single is there. And it consists of, this scutellum consists of plumule and the radicle. The plumule is surrounded by a protective sheath which is called coleoptile. Radicle is surrounded by a protective sheath which is called coleorhiza. The surface layer of scutellum contact with the endosperm is called epithelium. The function is to digest the food material stored in endosperm for growing of embryo. Okay, so you have to draw both the diagram in your copy as well as label all the parts. Okay, now next we have to study is the dicot seed, that is a bean seed. Bean seed is dicot because it contains two cotyledons. And what are the different parts in a bean seed that you can see? Number one, it consists of seed coat, and it is made up of two distinct layers, that is testa and tegmen. So if I draw a bean seed here, this outermost layer which I will draw is known as testa, and the inner layer is called tegmen. Alright? So this outer layer is called testa, this inner layer is called tegmen, Tegmen. These are the two layers of this bean seed coat. Okay? Then next we have is the seed coat protects the delicate inner part of the seed from injury and from attack of bacteria and insects. That is the function of seed coat. Both of them are part of seed coat. So if asked what is the function of seed coat, it protects the seed from bacteria and from injury. Now second point, hilum is a white oval scar. You can see in your book also, a oval scar I have drawn here. It is known as Hilum. Hilum. Now what is the function of this white oval scar called hilum which is present in the seed? It is present in the concave side. Like if I draw a seed, this one is concave side, this one is convex side. The bulging part, this is convex side. Okay, and this one, the one which is depressed inside, this is called concave side of the seed. Concave side. Got it? So hilum is a white oval scar which is present in the concave side of the seed, and micropyle lies at the end of this seed. It is situated close to the hilum. So if I mark a round circle here, that is called micropyle, Micropyle. Now what is the function of this micropyle? It is a tiny pore which is present in the seed, through which the seed can take oxygen during germination and it can breathe out carbon dioxide. Not only that, it is through this micropyle that the tube nucleus of the male gamete, it enters through the cells, through the micropyle only inside the female, what we say, embryo sac. The micropylar end is there, through which the male gamete will enter inside the ovule and cause fertilization or the discharge of male gametes. This micropyle will also help in the process of respiration of seeds. So micropyle, it marks the opening through which the pollen tube had entered the ovule. It also helps in the diffusion of respiratory gases. And one more function of micropyle is there, that when the seed is germinating, water molecules will enter inside the seed through this tiny pore only, okay, through the micropyle. Now next we have the embryo. Embryo is the baby plant which is present inside the seed, and it consists of, like, the plumule is there, which gives rise to the shoot system that produces the leaves and all. Then we have the radicle, which gives rise to the root system will develop in it. So this is called radicle, this one is called plumule, the one which is present inside the embryo. Now hypocotyle and epicotyle is, the hypocotyle is that part, hypo means below, cotyle means cotyledon, so that part of the embryonal axis which is present below the cotyledons. Now what are cotyledons? In a bean seed you will find that there are two cotyledons. Two cotyledons are there, that's why it is called dicot. And it is bean shaped or kidney shaped, as you can see from the diagram. They protect the embryo and store food for the growing embryo. Now next we will study about the differences between monocot seed and dicot seed. I will just say the differences. Number one, monocot seed contains only one cotyledon, whereas dicot seed contains two cotyledon. In monocot seed, there is plenty of endosperm present, so it is called endospermic seed. In dicot, there is no endosperm, so it is called non-endospermic seed, and if present is very less. Number three, monocot seed like maize had a very small embryo, but in case of dicot, the embryo is quite big in size. Then fourth one, plumule and radicle are protected by coleoptile and coleorhiza in case of maize seed, that is in monocots. In dicots, I have not drawn plumule and radicle. It means they are not covered by any type of sheath or covering in case of dicots. And the last point, in case of monocots, the hilum is invisible and it has a micropyle. So in monocots you can see the hilum is not only there, I have not marked any hilum, but in dicot like maize seed, the hilum as well as the micropyle, both of them are quite clearly visible. Okay, so now next we have to study is germination of seed. What is the meaning or definition of germination of seed? The process of change of a seed into a seedling or a baby plant in the presence of air, water and suitable temperature is called germination. For each and every seed to germinate, like if you grow any seed, sometimes it may not germinate or fail to germinate. Why? Because the seeds must undergo a period of rest, that is maturation. They have to break their dormancy in order to achieve germination. Okay, so you can find that although the seed is quite healthy, free from disease, yet it fails to germinate. If this type of question is asked, what should be the answer? Because the seed needs some time in order to mature, in order to develop itself and get ready for the process of germination. Okay, now next, what are the conditions necessary for seed germination? There are different conditions required. It's important to understand what is the use of all these conditions. Like, seed requires the following three conditions: oxygen, water and favorable temperature. I will just rub it. The conditions needed for seed germination are oxygen, water and suitable temperature. So let's see what are the uses of all these three factors, how they help in the process of seed germination, and if they are not present, what will be the effect. So first one is germination of seed. The three conditions which are required: oxygen, then we will study about the water and suitable temperature. Okay, what is the use of oxygen in germination? Answer is, seeds require oxygen to produce energy because germination needs energy to grow the shoot system and the root system. For the process of germination, energy is required, which is brought about by the process of respiration that is done in the presence of oxygen. So answer is oxygen helps to produce energy by respiration, because you know that food is already present inside the seed in the endosperm as well as in the cotyledons. So those food materials will come in contact with the oxygen and carry out the process of respiration to produce energy, and that energy will be needed for germination. Okay, so embryo needs energy for its growth, so it helps to produce energy by respiration for growth or for germination of seeds. So first condition you understood. Why do the seeds need oxygen for germination? Answer is to get energy, because energy is required for the process of germination, for the growth of different parts within a seed. Second one is water. Why the seeds need water for germination? One point is to make the seed coat soft. To make seed coat soft, because in the presence of water, the outermost hard covering of the seed coat, it will become soft once it is wet with water. The second function of water is needed for the process of, you can say, like, if water is not there, how can the plants carry out the process of photosynthesis also? But here in case of seeds, water is required because the concentrated food is converted into liquid form to change the concentrated food into dilute liquid food. To change, because you know that food is stored in the form of endosperm, which is solid, concentrated. To change concentrated food, I am writing only short, food into liquid form, because in the presence of water, that food will change into a liquid form that can be easily utilized by the seeds for the process of respiration. And water is needed for germination, for liquid form to be utilized by the seedling. Besides, seed coat becomes soft and get ruptured easily after absorbing water. To make the seed coat soft, for breaking the seed coat, because during germination the seed coat must open so that the baby plant can come outside from it. Okay, and the last one, naturally the seed contains water from the soil, which is absorbed through the micropyle. So food is stored in the concentrated form inside the seed. To change that concentrated food into liquid form, water is needed. The second function of water is to make the seed coat soft and to break the hard seed coat so that baby plant can come outside. Okay, so you should learn the points. And the last one is temperature. Temperature also affects germination, because if the temperature is 0 degree, the plants will fail to germinate or the seeds will fail to germinate. If the temperature is above 48 degree, more than 48 degrees C, then also no germination. But the seeds need an optimum or suitable temperature, which is between 35 to 40 degrees C. This range of temperature is good for germination. Okay, now the reason is why higher or lower temperature is not suitable for seed germination. Because at lower temperature or higher temperature, one by one I will say, at a lower temperature the enzymes are inactive, they will not work properly, and for the process of germination respiration is required, which is a enzyme catalyzed reaction in which enzymes are needed. Those enzymes will fail to work at a low temperature, they will not get activated. At higher temperature also the enzymes are killed or denatured, and that will also prevent the process of respiration, and thus the germination of seed will fail. So higher temperature destroys enzymes which are necessary for germination. Okay, now next we have to do is types of germination. 2 experiments are there that you can do later on. First we'll do the types of germination. Okay, Okay, so next is types of germination. I have just marked two types of germination are there, apigeal and hypogeal germination. What is the difference between them? Apigeal germination is the one in which the first point, hypocotyle elongates. Hypocotyle elongates. And here the first point will be apicotyle elongates. Apicotyle elongates. Okay. The second point is, that is important point. In apigeal germination, cotyledons are pushed above the soil. Cotyledons are pushed above the soil. Means cotyledons come above the soil during apigeal germination. Here you can write cotyledons remain below the soil. Cotyledons remain below the soil. And next one you can do is the example also you can write that this type of germination, that is apigeal germination, is found in the dicot seeds. So apigeal germination is found in bean seed or dicot, so you can write. And here hypogeal germination is shown by the monocots like maize seed. And the diagram is also there, figure 5.5 and 5.6, you can write them. And then even the steps are also written there, like germination of bean. The radical emerges from the ground, from the micropyle, in the direction of soil, water and gravitational force. Radical is the one which gives rise to the, like diagram is shown like this here, that this is the seed, then radical will emerge, that radical will grow downward, that is towards soil, towards water. Due to gravitational pull it is growing downward. Then this radical will germinate and it will develop into the primary root, secondary root, and then you can see that there is the formation of plumule also, that grow upward and then bent, that is arc downward. Okay, that these all are radical only. Okay. Then in the next diagram you can see that these cotyledons are pushed above. Now the cotyledons are here above, whereas the radical grow downward and the root system is being produced. So in this diagram you will find that it is the elongation of apicotyle, this part elongates, which pushes the cotyledons above the soil. So stepwise it is given. Number 2, radical forms the primary root, then the seed absorbs more water and it will swell up. The next step, the hypocotyle elongates or grows. So hypocotyle is shown here, this part as hypocotyle. In the diagram you can see, as well as here also it is marked hypocotyle, this part. So stepwise it is shown that hypocotyle will elongate and it will grow into a loop-like structure. This is called the loop-like structure. Now the hypocotyle is connected to plumule at the other end, which gives rise to the shoot system, and the primary root produces the secondary root. The secondary root will form the tertiary root, and that will result in the formation of a taproot system. The hypogeal grow and starts becoming straight and erect, and finally in the next diagram you will find that two cotyledons are there, and then there is the shoot system and the root system also develops. Okay, cotyledons are going above. Okay, and then the leafy structure is also formed. So this type of germination is found in this dicot seeds, whereas in maize seed also the diagram is also shown. In hypogeal germination, the cotyledons will not come out of this soil and apicotyle elongate. It will push the plumule out of the soil. The plumule will come outside, but these cotyledons will remain inside the soil only. Okay, and their steps is also shown. Now next section exercises are there. After that, one last topic, seedling. What is the meaning of seedling? Whether the germination of a seed is apogeal or hypogeal. The process of germination is change of a seed into baby plant, which is called seedling. And seedling is a small plant which is dependent on the food that is stored inside the seed, because that baby plant needs food to remain alive, to carry out respiration, to produce energy. Okay, the roots of the seedlings absorb water from the soil, and with the help of its roots, whereas the leaves of the seedling will make food by the process of photosynthesis and get the food, and the seedlings will keep on growing, like this is the baby plant or seedling, and it will germinate into a complete plant. So section exercises are there, like fill in the blanks. Number 1, a seed having 2 cotyledons is called, on page 48, it is called dicotyledonous seed. Number 2, the growth of seeds inhibited at very dash temperature, at very high temperature or low temperature. Number 3, the inner layer of seed coat is called tegmen. 2 layers are there, outer layer is called testa. In dash germination, the cotyledons brought above the ground. Above the ground means it is epigeal germination. And the last one, the maize grain shows dash germination. The answer is hypogeal germination. Okay, so you can try the review questions yourself. If doubt, you can ask it. Okay, and after watching the video, you can give your comments, like, and now there are 2 experiments also. I will just show you that experiment. You have already done in lower classes also. One is called 3 bean seed experiment, in which we take a beaker and on a glass slide you know, we keep 3 seeds at 3 different positions. Okay, we keep one of the seeds here, tie them, one in the middle and one inside at the lower end, and here water is present inside the beaker. Now it is asked that this seed is getting sunlight, it is getting air also, but no water, because it is, the water surface is only up to here. So this will not germinate. This seed, it is in contact with water also, like half of the seed is immersed inside the water layer is up to here. It means seed is getting water also, it is getting air also, and sunlight also, everything. All the 3 conditions are met here. So this seed, the one which is in the middle, will germinate, and this seed is fully immersed inside the water, so it is getting water, it is getting sunlight also, but no air. Air is not there, so this seed will also not germinate. So this is called 3 bean seed experiment. From this experiment, what is the conclusion we draw? We draw the conclusion or the result that for the process of germination, a seed will require all the 3 conditions of air, water and sunlight. Okay, now next we have to do one more experiment is there, that also I will draw and then explain it. This experiment can be asked in the questions also, so be careful. Diagram based questions or experiment based questions can also be asked in your examination, board exams also, because you know that you have to write the questions which are given by the board exam, not by the school. So now this experiment is to show oxygen is necessary for germination. What is the aim? The aim of this experiment is to show oxygen is necessary for germination. Okay, now what is the experiment and how it is done? There is a conical flask. You know what is a conical flask? Okay, here is a stopper is kept, and then inside this conical flask, with the help of a thread, we suspend a small test tube in which a chemical is kept. That chemical is, the name of that chemical which is present, alkaline pyrogallic acid. Alkaline pyrogallic, only one name it is, not different names. Okay, pyrogallic acid. What is the function of this chemical? And there you can find wet cotton is kept on which the seeds are placed. Maize seed, gram seed, anyone. Here also in this experiment also we do the same. I will make it somewhat bigger so that you can understand. Okay. Okay, now see. This one I have made it so thick, so big. This much big is not required. Otherwise it will change the shape of your conical flask. Here the stopper is there. Okay, there is a cotton swab. Cotton we have kept, which is wet, so that it can help the seeds to germinate. Okay, and we have kept the seeds. Now we have also introduced a test tube inside it, but this test tube contains only water, plain water is there, nothing else. In this test tube we have kept plain water. Okay, this one is experimental setup number A, this one is setup B. Okay, now after keeping this apparatus for a few days, we have to compare and see the result. You will find that the seeds which are kept in flask B, it will germinate. It will germinate. Why? Because the seeds get water from this wet cotton swab, which is kept at the bottom of the conical flask. This is the flask. This is the stopper. Okay. Now inside the flask we have suspended a small test tube containing plain water. So for germination, what is needed? We need water is there, sunlight will be falling on it, and water, sunlight and air is already present inside it, because it is trapped inside it. Even the water is also there, so air is already present inside, so it will show germination. Okay, now in flask A there is no germination of seeds. Seeds will fail to germinate. Why? Because the function of this alkaline pyrogallic acid is to absorb oxygen. Whatever oxygen is present inside this flask, it will be absorbed by this chemical. Means there is no oxygen inside this flask. It is already absorbed inside this alkaline pyrogallic acid. So this seed will get the water, it will get the sunlight, water, but no oxygen. No oxygen means no germination. So from this experiment, we conclude that oxygen is necessary for germination. Hope you have understood. Okay. Now I have told you that if we'll get some time, we'll discuss the previous chapter question answers. Even though if you have, you can try yourself. If doubt, you can ask. Like on page number 41, select the correct option is there. Number 1, the male part of flower is called stamen, pistil, stigma, embryo. Stamen, A is correct. Number 2, after fertilization, ovule develops into seed. Number 3, the fusion of one male gamete with an egg, the other with secondary nucleus, is termed double fertilization. Then number 4, wheat and maize both are insect bud pollinated? No. Insect pollinated? No. Wind pollinated plant? Yes. Okay, because maize is wind pollinated. Bisexual flower which do not open at maturity, they are called cleistogamous flower. So you read your book, you will get all the answers. It's not so hard. Even if you have doubt, you can ask. Okay, thank you.